Good morning. Thank you for resisting the temptation to go on the science walk this morning. It's a hard choice to make. My presentation this morning and my role on the Scientific Advisory Board was really to look at the rigor of the systematic review process itself. And so my disclosures here that influenced my thinking, a decade at the Academy working with the Evidence Analysis Library, and potential for the funding from the ILSI to be on the Scientific Advisory Board. So the role I had was to look at the systematic review process throughout the whole planning and conducting and look at how we were meeting the standards. In this particular case, my presentation is going to go through the standards that were published by the Institute of Medicine and kind of demonstrate for you the details for how this systematic review met those particular criteria. And then Danielle is going to come back and circle around and give you the details of what it felt like to do the different activities that are included. We all know that the goal of a systematic review is to summarize or synthesize the best available research to inform decision-making. Most common purposes are to create treatment guidelines or practice guidelines, identify gaps in research, and provide input to policy decisions. When we think about systematic review, it kind of emerged in the medical field about the 1990s. About 1995, JAMA started the series on evidence-based medicine. In nutrition, right around 2000 is where it really started to build impetus in nutrition. The Evidence Analysis Library at the Academy came online 2004. Then 2009, the Nutrition Evidence Library came online to support the dietary guidelines. And more recently now, the OHAT in the toxicology world, their first guidance was provided in 2013 and 2015. So this review is relatively new to the toxicology field. The same way as the prevalence of the systematic reviews, then also the guidance has been evolving. There's guidance for how you publish systematic reviews. The most recent one is 2009, the PRISMA guidelines. And there's also guidelines for how you conduct the reviews. And again, it just shows the time frame, and that the toxicology one was first draft published in 2013, and then the final guidelines for that 2015. For our particular review, we selected these four guiding principles or guiding documents. Finding what works in healthcare, that's the standards I'm referring to for systematic reviews. We used the OHAT methodology for risk of bias. For synthesizing the body of evidence, we used the GRADE methodology. And then for publishing, we conformed to the PRISMA. So I'm going to walk you through the standards. Most of you are probably somewhat familiar with them, but I'm going to show each of the standards. There are 4 chapters, so there's 4 different standards, and this is the same way that the whole review was organized. So you'll keep seeing these same themes throughout. There's 1 chapter on how you do the protocol with standards for that, 1 standard for how you find and assess the individual studies, 1 chapter that deals with standards for how you synthesize the evidence, and then 1 chapter that deals with reporting systematic reviews. So the protocol development chapter is the longest, and these are the different criteria that are included in that particular chapter. So the first one is establishing a multidisciplinary team, and the IOM report specifies the expertise that is needed. You need to have relevant clinical areas, systematic review methods, a person who's qualified and trained in searching for the evidence, a statistician for the quantitative methods, and then other expertise is needed. In this case, the team had 2 major components. There was the scientific review team that did the work at Tox Strategies, and then there was a scientific advisory board that complemented them with the clinical expertise and the systematic review. The expertise for the group from the Tox Strategies are listed here. You'll see that there's expertise in food safety, toxicology, epidemiology. People had been involved in doing literature searches before and certainly had quantitative methods and statistical background. In addition, as we were planning, we realized we didn't have a systematic review trained librarian on the team, and so that was in fact added as an additional consultant. And Tox Strategies also had a biostatistician that's not listed here but was available for consultation during the systematic review. When you look at the Scientific Advisory Board, this is where your clinical expertise areas come in. So we have cardiovascular, physical performance behavior, nutrition and behavior, reproductive and development, pharmacology, and bones and calcium. And then my role was the systematic review person on the Scientific Advisory Board. Since it was funded by industry or by ILSI, everyone always wants to know, so what did ILSI have to do with this? So the ILSI's role was to establish the scope of the project, and that was established to identify the need by the ILSI Caffeine Working Group. And they also identified the need to make sure that we met the IOM standards throughout the process. They selected the vendor, and then they provided the resources. And throughout the course of the systematic review, the only time that they were consulted was when we needed to have additional resources. So, for example, the librarian wasn't in the original project, and so we went back and said, we really need to have this added, and then so they were able to provide the resources to do that. They coordinated the teleconferences in terms of the mechanics of it, and they facilitated the stakeholder input from FDA and Health Canada at specific times throughout the process. But be assured that at no time did they provide any input on the actual process itself or the content that was included in the systematic review in terms of influencing those. The second standard, there are two standards dealing with managing bias and conflict of interest. So both the Scientific Advisory Board and the Tox Strategies team followed the plan that had been established. We declared conflict of interest 2 times, at the beginning of the project and then just before the manuscript was submitted. The disclosures were made in the group teleconference, and they're also included in the published Prospero protocol. The way it was managed in calls and meetings is you were to self-declare if there was a situation where you felt you had conflict of interest and to withdraw from the discussion. Ensuring stakeholder and user input was facilitated by ILSI. For example, they met with FDA and Health Canada at 3 different times during the project. At the beginning, when the scope of the project was being developed, after the first list of articles had been identified, and after the initial synthesis was done. So the type of input that was provided back to the group from FDA were things like, yes, it's really important that you look at children. Please identify research areas. So that's the kind of input that came back to the Scientific Advisory Board. The Scientific Board met by teleconference 4 times where all of us were on board. And then in addition to that, there were endpoint calls with the Scientific Advisory Board members at the Phase 3 and Phase 4, and there were, as you can imagine, multiple emails soliciting input in the different phases of the project. There's a standard saying, did you properly formulate the topic for the systematic review? And we used the PICO format, so population exposure was used instead of intervention, and then comparator, and we mentioned that NAROT was our comparator, and then the outcome. There were 2 different levels of questions. There's the overarching question about what we know about safety of caffeine levels, but then there were specific sub-questions dealing with specific populations, with specific health outcomes, and with specific endpoints. The topics that were identified by the Scientific Advisory Board and Tox Strategies were eventually divided into 2 categories. One category were the things that were included in the formal PICO questions in the systematic review, and this dealt with those 5 broad health outcome areas. And there were 27 specific endpoints and 4 subpopulations. The one item that was pulled over to be included as background information, data collected but not formulated in a PICO question, was the information about pharmacokinetics. The analytical framework is shown here, and Danielle will go through and walk you through the specifics of it, but it identifies what the population was, what the comparator was, what the exposure measure was, and the major outcomes. There's a criteria about identifying what needed to be included in the systematic protocol. So all of these data elements here were to be included. The background, our colleague from FDA has told us about NAROT. That was the background, highly cited reference, but it had been a long time, and it hadn't been done using the systematic review process. Now, to show you what goes into the protocol, I pulled one of the protocols submitted to Prospero, and this is the one for reproductive and development. So I'm just going to go through and give you an idea of the level of detail that was included in the protocols. It identified the types of studies to be included. It identified the condition or domain that was being studied. It identified the participants and the population. It identified more specifically what was included as a healthy population and what was included as excluded as an unhealthy population. It talked about exposure, what kinds of measures were to be included as in the measurement of exposure to caffeine. and it talked about what things were going to be excluded, and these were specific to each protocol. So for example, this one talks about excluding studies that assessed caffeine exposure to the pregnant woman after the pregnancy. That was not included, for example. The outcome measures were identified. The primary outcome measure dealing with adverse reproductive and developmental effects, and all in the times we're collecting information on the pharmacokinetics as a secondary outcome. This is an example of the 10 endpoints that were in the reproductive and development health outcome. We also needed to identify the time point for both the exposure, which was during pregnancy, and the outcome, which was during pregnancy, at birth, or after birth. The exposure was the total daily caffeine intake, and then the comparator was pregnant women or children of women had a daily intake of the NAROT level of 300 milligrams of caffeine during pregnancy. There was a criteria dealing with the search strategy. The IOM recommends you have more than one database, and it be conducted by someone who is a systematic review trained librarian, and all those data elements are in fact included and published. Procedures for study selection included, did you adequately identify how you were going to do this? The titles and abstracts were compared to the criteria, the inclusion-exclusion criteria, by two people at Tox Strategies. They were classified as either to be included, excluded, or uncertain. And if there was anything that was uncertain, then they were discussed by additional two people from Tox Strategies. At each step, they were reviewed sent to the clinical expert on the scientific advisory board to look at the decisions that were being made. After the initial decisions, then the full articles were reviewed, and the same process followed. The data extraction strategy needs to be described in the protocol, and this was described as the data was being extracted into its distiller SR. The data from there then were downloaded and provided to the scientific advisory board, and in the end, the data is all uploaded to the SDSR, the systematic review database from AHRQ, and publicly available. The data extraction strategy identified common data elements as well as endpoint-specific data. And there was a data extraction plan. They pilot tested all the forms, and the whole goal was to provide a very high level of transparency so anyone could see exactly how it was done. The process for identifying and resolving disagreements was usually conducted through email and teleconferences. In most cases, it was between the Tox Strategy staff, the scientific advisory board members, and I was on board to facilitate that. We always had the possibility of requiring or documenting a minority opinion, but luckily we were able to reach consensus on everything, and this was not required, but the process was there. You need to identify how you critically appraise the individual studies, and in this case, the OHAT risk of bias tool was the one that was used, and these are the 11 criteria that are included in that particular risk of bias tool. You need to describe the method for evaluating the body of evidence, and we use the grade methodology, where you increase or decrease the confidence based on the criteria that are listed there, the considerations, level of adversity. And you end up with the body of evidence being classified as either high, moderate, low, or very low. In further describing how to evaluate the body of the evidence, there were two methods that were used. One was creating tables of data, and the other one was to include narrative information in the chapters and conclusion statements. The conclusion statements are at two levels. There's one conclusion at that individual endpoint level, and then there is an overall conclusion statement at the 5 topic areas for the health outcomes. So for example, in reproductive, there were 10 individual endpoints, so there were 10 conclusion statements at that level, and then 1 for the overall reproductive. You need to describe what kind of analysis for different subgroups, and in this case, the PICO statement and analytical framework included 4 populations: healthy adults, healthy pregnant women, healthy adolescents, and healthy children. And you need to specify a timeline, and this timeline is organized, and you'll see this multiple times according to those 4 phases: the protocol development, assessing, synthesizing, and reporting. On the left-hand side, the gray arrows show where the scientific advisory board provided key input, and on the left-hand side where you have the pink and the blue, this is where the input came in from other stakeholders like FDA and Health Canada. And then finally, you need to submit your protocol for peer review, and all 5 protocols were submitted to Prospero. So there's a separate protocol for each of those 5 health outcomes that has been submitted to Prospero. The next chapter deals with how did you find and assess the individual studies, and these are the criteria that are included. You need a comprehensive systematic search for evidence, and this was completed using the protocols. It was reviewed, and there were no additional resources added or articles added. Take action to address potentially biased reporting. In this case, because there was a huge body of research, we were not able to do the IOM recommendations of searching gray literature or asking researchers to submit unpublished data, hand searching or web search, and we did limit to the English language. It was just not, there was a huge body of research. Criteria for selecting and screening the studies. The protocol was tested prior to use of it. All the responses were documented in a screening form and uploaded to Distiller SR. They were reviewed at the title and abstract level as well as the full text review. And the IOM recommends including observational research in addition to RCTs when you're addressing harms, and this was done. And then in terms of documenting the search, we mentioned that the search syntax is all uploaded as an attachment to Prospero, and the results of the screening line by line are loaded into Distiller. In addition, the manuscript includes the diagram that explains how many articles were at each step, and the SDSR has the line by line. And this is the reference, so you can go look to each of the proposals. In terms of managing the data collection, the requirement is to train and pilot test using the forms. The Distiller software was used for the data collection. The reviewers worked independently. The standard data collection form for each health outcome, for each population, and for each endpoint. It included the data that the authors had in the original research as well as data that the extractor had to compile. And there was a second level of review data were reviewed by the Scientific Advisory Board through emails and teleconferences. This gives you an example of what I mean by basic information from the author, and on the right-hand side is the customized fields, for example, that they would have had to create from the data that was in the research project, like standardizing the exposure dose. When they looked at critically appraising the study, they used what they call a heat map. It's color-coded, and across the top are those 11 criteria from the OHAT risk of bias tool, and Danielle will go through this in detail with you. In terms of standardizing how you synthesize the body of evidence, we use qualitative synthesis. In addition to forest plots, the narrative conclusion statements, and tables showing the confidence of the evidence, and then the final summary for all the endpoints. So for example, the plots were displayed using colors and symbols and sizes of symbols to represent the data ranges and the quality of each of the individual studies included. The confidence body of evidence table included the initial confidence rating. It showed very clearly the impact of the risk of bias rating. It showed the impact of the other grade criteria, and then the final confidence rating. The overall summary table included information like the outcome, the endpoint, comparator population, the number of studies that were included in the range of the data. It showed a decision whether the comparator was either too high, acceptable, or too low. It included the level of adversity in terms of types of effect and level of confidence, and finally included the overall conclusion statement and the overall comparator conclusion. When you conduct a qualitative analysis, we also created the conclusion statements that included these components. So each conclusion statement had to identify the population, the number of studies, the spectrum of the endpoint conclusions, the level of adversity, and the outcome, or the overall level of confidence. So this is actually what it looked like. This is a conclusion statement at the endpoint level. So this is talking about recurrent miscarriage. So the items that are highlighted in yellow are the things we're talking about. There were 4 studies, moderate level of confidence, acceptable intake, recurrent miscarriages was the condition, healthy pregnant women was the population, confidence was increased by a level of indirectness and low risk of bias and reasonable consistency. When you look at a conclusion statement at the health outcome level, it also includes the number of studies. In this case, it was 58 studies. It describes the studies, how many were observational, how many were trials, health outcomes, and populations, identifies, again, at the very end, this is a moderate level of confidence. So it gives you an idea how they were constructed. There was a decision not to pursue meta-analysis. It was not necessary to answer the question that we were looking at. The data is all there, so if someone wants to go back and do meta-analysis, they would need to be done at that 27 endpoint level is how you would do the meta-analysis for that. The last criteria talked about standards for reporting. The final report recommended by IOM includes all of these components. The only two that are not yet completed is the executive summary and the summary for the lay public. It recommends a peer review of the draft report. The manuscript was submitted, and the peer review was conducted by journal reviewers. We also had review by the Scientific Advisory Board as well. And then publish in a manner that ensures free access, and the article is published, they mentioned yesterday, right? Yesterday, it's open access to everyone. The SDSR includes all the data, and then the protocols are on Prospero. So, now you know how we met all the standards. Now I'm going to turn it over to Danielle, and she will tell you what it really felt like to do the review and the challenges and then the specifics of it.